Assalamu alaikum. Hello everyone. My name is Aalaa Albasha, first year PhD student working under supervision of Professor Galvaraz Khan, College of Medicine and Health Sciences, United Arab Emirates University. And today I'm going to present our project in which we use a novel rabbit model of Epstein-Barr virus infection to understand the dynamics of EBV-induced lymphoproliferative disease in immune-compromised status. My presentation will consist of brief introduction, methodology, results, and conclusion. Despite notable development in transplantation and immunosuppressive therapy, post-transplant lymphoproliferative disease remains one of the most feared complications following solid organ transplantation. At the center of this problem is Epstein-Barr virus, or EBV, a double-stranded DNA virus belongs to herpes viridae family. EBV is strongly associated with wide spectrum of cancers, such as gastric cancers, nasopharyngeal carcinoma, and wide range of lymphoma, such as Burkitt lymphoma, Hodgkin lymphoma, and post-transplant lymphoproliferative disease. EBV virus enters the human body through the oropharynx. Then it infects the resting B lymphocyte. After infection, it establishes several types of latency program with expression of different viral protein. This latency enables the lifelong persistence of the virus in the body and evading of immune surveillance. Reactivation takes place under circumstances such as immune suppressive status. Understanding how this process begins or how the EBV virus induced lymphomagenesis is still poorly understood, probably due to a lack of suitable in vivo model. To address this, our lab has developed a novel rabbit model of EBV infection. This model allows us to study the virus under controlled experimental setting. In this experiment, we use a white New Zealand rabbit young age, 3 to 5 weeks. We collect the blood sample, pre-treatment blood sample, as the baseline for our experiment. Then we divided the rabbits into three groups. The first group is a control, healthy control and infected, and the second group is EBV infected immune competent group. The third group is EBV infected immune suppressed group. For this group, the third group, we use a daily injection of cyclosporin A to mimic the scenario of post-transplant immune suppressive status. We followed up the rabbit and we collect the blood on weekly basis. Sacrifices took place at different time points and we investigate the organ's abnormality at the gross examination level to see if there is any structure or features that is not normal. We collect and harvest the major organs and preserve it for downstream analysis, which include the molecular techniques and histological techniques. Moving on into results part, which is consist of vital and gross examination, histological findings, and molecular findings. And I'm going to start with vital and gross examination. As I mentioned earlier, that we followed up the rabbit by measuring the weight so that we can know the growth rate of each group. So we notice that there is a significant decrease and reduction in the growth rate of the immune suppressed group compared to the other two groups, immune competent and healthy control group. At the gross examination of spleen sample, we notice that there is a nodular structure in the spleen sample from immune suppressed group. Also, there is abnormality in the feature of the spleen itself when we compare it into immune competent and healthy control group. This abnormal feature is further investigated by histological techniques. We perform hematoxylin and eosin as the primary microscopical analysis to see the feature of the tissue. On the spleen sample from the immune suppressed group at different time points, we notice that there is a destruction in the normal splenic architecture. We wanted to investigate, is this abnormality related to virus? Does EBV involve in these changes and pathologically? So we did the in situ hybridization for detecting and localizing EBV encoded RNA, and we found there is a heavily positive result in this abnormal structure or abnormal part of a spleen sample. We further perform immunohistochemistry to detect one of the most important virus proteins that related to oncogenesis, which is latent membrane protein 1. And we found there is a heavily expression of this protein in the spleen sample. Furthermore, we want to detect the virus dissemination into several organs like liver, lungs, and tonsils. As we can notice here at H&E, we saw there is an infiltration of lymphocyte to the liver and the lung, even with alteration of the normal structure and feature of the lung. This infiltration was happened by infected lymphocyte, as it appeared as positive with both hyper-IgE and immunohistochemistry for latent membrane protein 1. Interestingly, we found there is several cases of early signs of lymphomogenesis. In these cases, for example, which I'm presenting today, there is a well-demarcated area of lymphoid lesion in the spleen sample of immune-suppressed group. This area is surrounded by collagen fibers, as it appears as blue color when we did trichrome stain, and also it is induced by EBV, as we did in situ hybridization for EBV detection and immunohistochemistry for LMP1. Immunophenotyping of this lymphoid lesion revealed that the vast majority of this cell is CD79A positive, which mean B lymphocyte, with the minor presence of CD3 positive, which is T lymphocyte. At the molecular level, we wanted to quantify the EBV copy number at different types of tissues and organs, such as blood sample, spleen, and liver. We noticed that there is a statistically significant increase in EBV copy number in the liver, spleen, and blood for the immune suppressed group compared to the other two groups, immune competent and healthy control group. This molecular finding, together with the histological finding, confirms that there is a dissemination of the virus into several organs. In conclusion, our study demonstrates that for the first time in a controlled experimental setting, that EBV under immune suppression is associated with pathological change reminiscent of early lymphomogenesis. Our finding further confirmed that LMP1 seems to be the key driver of this pathological change. And finally, the rabbit model of EBV provides a robust and highly relevant in vivo platform for studying EBV-associated diseases and for testing potential therapeutic strategies in immune-suppressed patients. Together, this finding provides a valuable framework for understanding EBV-induced lymphomogenesis and for designing interventions aimed at reducing post-transplant lymphoproliferative disease. I would like to thank the College of Graduate Studies, United Arab Emirates University, and also Jalila Foundation Research for supporting this project, and for sure the Beyond Science Initiative for giving me the chance today to present my work in such an outstanding conference. This is our team, Professor Khan Viral Pathology Lab. Thank you all.